Buen día a todos y bienvenidos a la semana 2 a la clase Microbiology 205. Welcome to week 2 of Microbiology Lab. This is my first time sharing my video announcement with you because I was having some hardware trouble last week when the class started. But here I am. My name is Jonathan Jeffries, and I am your professor and guide as you traverse the world of, as I call it, microbiology. I call it the world of the unseen. Now, the big takeaway from last week was that microorganisms are ubiquitous, right? They are everywhere all the time. In every niche, on every continent, you can find microorganisms living and thriving there. The big takeaway from this week is that microorganisms are, well, microscopic. Okay, I know it's a kind of against convention to use a form of the word in the definition of the word. So how's this? Microorganisms are tiny. I mean, really, really, really tiny. Microorganisms are so tiny, in fact, that we can't see them with the naked eye. We can't see them with the unaided eye. And that's where the microscope part of microorganisms and microbiology and microscopy, that's kind of a cool word that really means seeing tiny things under a microscope. But that's where those words come from. Now, the use of glass lenses to view very small things evolved in the 13th century, believe it or not. And those lenses were more like our modern day magnifying glasses. History has bestowed upon Dutchman Antoine van Leeuwenhoek the honorary title of Father of Microbiology due to his observations of what he called animalcules, little microorganisms in pond water, using his handmade, simple handheld microscope. You should go to Wikipedia and look up Leeuwenhoek, and you'll see that little tiny handheld microscope. It's pretty cool. Now, that was in the mid 1600s. Well, fast forward to the 21st century, where even the simplest of microscopes, even the ones that children get for their birthdays or as Christmas presents, even those are dozens of times more powerful than even Leeuwenhoek's. So, one thing I hope that you will appreciate this week is just how small bacteria really are. So the example that I gave in my printed announcement is of E. coli. That's Escherichia coli. Of course, you know it better as E. coli. Is about 2 micrometers, or we say 2 microns for short. 2 microns long. Now, to put that into perspective, a human hair is about 80 micrometers in diameter. A micrometer is 1/1,000,000th of a meter. Too small to be seen with the unaided eye. A human hair, 80 micrometers in diameter. 2 microorganisms are, for the most part, colorless, and some are even transparent. And so it's for that reason that we use stains in microbiology, so we can view structures like cell walls and chromosomes and all those sorts of things. And that's what one of the things that this week is all about is microbiology stains, just chemicals that we plop onto a specimen that then gets taken into the specimen, and then we can see various structures. And that brings us to this week's objectives. This week you will learn to apply a simple stain to visualize microbial cell structures like cell walls and chromosomes. You'll learn about the Gram stain. Now, this is sort of the grandpappy of all microbiology stains, named after, of course, Dr. Gram, who invented the process. It's a differential staining technique where, and that's Gram stain with a capital G, named after, of course, the person. But it is based on use couple of different chemical dyes, and it's based on the cell wall structure of the bacterium. You'll hear this word peptidoglycan a lot. Gram-positive organisms have a very thick layer of peptidoglycan, and gram-negative cells have a very thin wall of peptidoglycan, and it is the thickness or thinness of the peptidoglycan layer in the bacterial cell wall that allows it to take in the gram stain or refuse the gram stain and to get counter stained or not. And so that you get some organisms appearing purple or violet or dark blue, and other organisms appearing red or pink. And that's how you differentiate gram positive. We call them gram positive and gram negative bacteria. That's kind of a first step when you are trying to identify an unknown bacterium. You do a gram stain, and that effectively divides your choices in half. If you've got purple, you know they're gram positive, and so you don't even bother looking at any gram negative organisms because it's not that. So that's a good way to sort of divide or to kind of label your organism, your particular unknown organism, into a category, which then you can focus on other tests to identify your unknown. Okay, you'll learn more about that as you get into later on when you actually, I think it's week 5, when you actually will do not a gram stain, but you will identify an unknown bacterium, and that's one of the tests that has been done for you is the gram stain to help get you started. Okay, and you'll learn about the acid-fast technique for visualizing endospores or spores. You'll learn more about that this week as well. All right, housekeeping items. Order your microbiology equipment kit. I think many of you have done that already, and some of you have actually contacted me asking me if you can get started early. And that's perfectly fine as long as you're following the correct instructions. But if you have not ordered your microbiology equipment kit, that comes from Carolina Biological Supply over here on the East Coast, North Carolina. And if you didn't do it first thing last week, make sure you get it done today so that you can have your kit. By the time we get started, we start using them on Monday next week, week 3. So make sure you get your kit. If you're not sure, just go to class resources. Second item down, how to get your microbiology kit. You don't have to pay for it. Just pop in your order and put in your shipping address, and they'll ship it to you. They're usually pretty quick, but a lot of people are ordering stuff, this being early in the new semester. So make sure you get yours on the way if you haven't already. Make your discussion post early in the week so that you may fully participate in the discussion forum. As we get in week 2 here, we're getting into more substance or more meat with regard to our discussion. Last week was like introduce yourself and what do you know about microbiology, and that sort of kind of the baby steps. And everybody, I usually give everybody credit for everything the first week. Now, as we get more into substance as far as the class goes, I have to start looking at, are you contributing to the discussion when you respond to a peer? Are you submitting a substantive response, 150 words, including at least one authoritative reference, properly cited? Like, I have to look at those kinds of things. So that's where some of the differential grading will come into play. But make sure, some of you like to wait until the last minute of the last day when something is due to turn it in. But if you wait until like 10:30 Friday night, right? Because the cutoff is 11:59 p.m. Friday for your initial discussion post, right? But if you wait until then to do your initial discussion post, then that doesn't give your classmates enough time to really read your post, digest it, and respond in a proper way. The sooner you get your post in, of course, I have to give you credit if you get yours in at 11:59 on Friday. But the earlier you get it in during the week, then that gives your peers time to respond, and you can really create a discussion. Right? The whole point in discussion is think about it like this: if all of us are in a room, this is a physical classroom. All of us are in a room, sitting around a big table, and I throw out a topic, pathogenesis of E. coli. And we go around the room, and everybody, you do a little research, and everybody pops in their ideas and their thoughts. And Sue says one thing, and then Jack comes back and says, Sue, yeah, that's a good point. What I found out is blah, blah, blah. And it keeps going like that. That's the way discussion is supposed to work. The earlier you get your initial post in, the better that process works. I stress a lot about APA. Okay, this is the American Psychological Association style guide for references and citations, and I stress a lot about that because the university stresses a lot about APA formatting, because when you go, this is a 200 level course, but when you go to 300 and 400 level courses, and some of you will go to graduate school, then you gotta know APA, especially in graduate school. It needs to be second nature. It needs to be like A, B, C, D, E, F, G. But now I have the book. You don't have to buy the book. There is a reference that the university has provided. It's in the student success section on APA. And also, if you're not sure, if you want to, you pull up a research article and you're not sure, you can just Google it. You can put in how to format a research article in APA 7. Always put APA 7 because this is the 7th edition, and you don't want to get the proper format from the 5th edition, right? So always put in APA 7, and it will tell you how to do that. On that line, I don't know, whoever, has not updated your textbook reference. So, end of last week, I posted an announcement on how to reference your textbook that is Nestor's Microbiology. Okay, so that's on there for you, and I have the format of the citation there as well. Okay, that's that. Let's see. Your initial post due by no later than Friday, January 23rd, 11:59 p.m. GCU time. We're working on topic 2, bacterial staining. You go to T2 or topic 2 and click on that. That opens up a drop-down menu where you will see your activities for the week. There is a pre-lab. Every week there's pre-labs. I call them pre-labs. Pre-lab questions for labs 4 and 5. This assignment is due no later than Sunday. That's January 25th, 11:59 p.m. GCU time. Start working on the lab activities. We had 7 last week. We have 5 this week, and you don't want to wait until Sunday to start working on them because it takes a little bit of time. Work on one today, or a couple today, a couple tomorrow, whatever. You can do them all at once if you want, but don't wait until Sunday because you probably are never going to get them all done. There are 5 of them. They are due no later than Sunday, January 25, 11:59 p.m. GCU time. With regard to grading, there is no integration between Canvas and Halo. So, McGraw-Hill Connect, and Halo. So, I have to manually transfer grades from McGraw-Hill Connect over to Halo. So, if you're looking at your grade book today, and you're seeing a bunch of like overdue. That's just because I haven't had time at this point to transfer grades. I'm on the East Coast, and right now we're on daylight savings time. That's 2 hours ahead of Arizona, all of the due deadlines are 11:59 p.m. Arizona time, or I'd say GCU time, which would be like 2 o'clock in the morning on the East Coast. So I'm usually not up at 2 o'clock in the morning transferring grades over, so I always do that. Try to do that on Mondays. So if you're looking at your grade book and see a bunch of overdue, don't freak out, and you don't have to email me or message me and say, Jeffries, what's going on here? I'm pretty sure I did my assignments. I just have to transfer grades over. Not an issue. If you do not like seeing that overdue on Monday morning, then you can post a screenshot of your completed assignment. You can post a picture of your dog or whatever. You can post something there just so just so Connect will read it as you've submitted your assignment. You'll see a blue submitted there and everything will be fine. But it'll be Monday um, at the earliest that I can transfer grades over, and that's when your gradebook will be updated. So thank you for your patience ahead of time on that. Okay. I will add an optional discussion question. If you complete it, it counts towards your participation points. Your response must be substantive, but you do not have to reply to colleagues if anyone else responds to my prompt. That response, though, is due no later than Sunday, January 25, 11:59 p.m. GCU time. I always say pay particular attention to due dates on all of your assignments. Proper credit can only be given for complete and timely submissions. If you have any questions or issues or problems, you can contact me as soon as possible. You can message me directly, 804-229-7993. You can message me on Halo, on the Halo inbox or message box, that platform there. Those are the two quickest ways to get a hold of me if you need me right away. You can also send me an email through campus email. I do check my email multiple times during the day and throughout the weekend, but not the quickest way if you need something right away. If I'm in my office and you message me directly or message me on Halo, I will usually get back to you within a couple of minutes, 30 minutes maybe, um, even though I have technically 24 to 48 hours, but I usually respond right away. So I did post a video about the gram stain, all right, from Neuro Academy. So check it out, YouTube video from Neuro Academy on the gram stain. You'll see the difference between gram positive, gram negative, and that will help you out in understanding some of the junk that you're going to be exposed to this week. With regard to the quiz, as I was looking over the quiz, oh no, wait. I'm thinking of a different. I'm thinking of lecture. All right, you guys don't do quizzes here. But anyway, be patient with me as I transfer your grades over, and let me know if you have any issues or if anything is going on. Get your kit, order your kit if you haven't done so, and if you have your kit and you're interested in kind of starting early, let me know so that I can pre-guide you so that you're not running down the wrong rabbit hole. Okay. That's all I have for now. Again, my name is Jonathan Jeffries, and yes, I got a tooth knocked out, and I'm waiting for my flipper, so don't let that scare you. Talk to you later, and have a good week.